So chapter 6 is our first chapter in Unit 2. In this chapter, we're going to talk about metabolism, energy, thermodynamics, endergonic and exergonic reactions, work a little bit more about ATP, and then enzyme stuff. So metabolism is the sum of all of the chemical reactions carried out by an organism, and metabolism really requires enzymes to occur in a timely fashion. We'll talk more on enzymes coming up here in the next couple slides. Broadly, we can talk about anabolic pathways or anabolism and catabolic pathways or catabolism. So anabolic pathways synthesize larger molecules and they require energy to do so. For example, linking amino acids together to build a protein would be an example of an anabolic reaction. This is kind of cheesy, but I put some dogs up there lifting weights because many people are familiar with anabolic steroids. I'm not suggesting you take anabolic steroids against medical advice or anything like that, but sometimes people take anabolic steroids to build muscle. So we've got those dogs up at the top lifting weights to represent anabolic reactions. Catabolic reactions break down organic molecules. So we've got this cat picture at the bottom where this cat has broke a flower pot, knocked it over. So catabolic reactions break down organic molecules, and when they do that, they release energy. So cellular respiration, which involves the breakdown of glucose and other organic molecules to release energy, that would be an example of a catabolic reaction. So energy is the capacity to cause change, and living things need energy in order to maintain order and structure and stay alive. You've maybe heard of kinetic and potential energy before. Kinetic energy is the energy associated with motion, and it includes thermal energy, which is kinetic energy that's associated with the random movement of atoms and molecules, and heat is thermal energy in transfer from one object to another. Potential energy is energy due to structure or location, and an example of potential energy includes chemical potential energy, which is energy that's available for release in a chemical reaction. So a large molecule could be broken down into a smaller molecule, releasing energy. So the energy in those molecular bonds is a form of potential energy. So thermodynamics is the study of energy transformation, and honestly, there's whole classes on thermodynamics, but broadly, in an open system, like an organism, energy and matter can be transferred between the system and its surroundings, and living things follow the laws of thermodynamics. The first law of thermodynamics says that energy can be transferred or transformed, but it can't be created or destroyed. That's the principle of conservation of energy. With this picture with the bear here, so there's energy in the fish, and the bear is eating the fish, and it's transferring the chemical energy from the fish into chemical energy in the bear. The second law of thermodynamics says that during every energy transfer or transformation, some of the energy is transformed to heat. So it's not truly lost, but it's lost from the organism to the environment. So every energy transfer actually increases the entropy of the universe. In this picture here, the bear is taking its stored chemical energy, which is a form of potential energy, and it's running. Like, look at how fast that bear is probably going. Like, the water's splashing up everywhere, right? That bear is moving. So some of the energy that was stored in the bear's fat has been released in order for that bear to have the energy to move, and the bear is also releasing heat from its body, because not all of the energy stored in the bear's organic molecules are causing the bear to move forward. Some of that energy is not transferred to the bear's movement, it's instead transferred to the environment as heat. So let's review here. As always, pause if you need to. So the first one says a diver has blank potential energy on the platform than in the water. So the diver is actually going to have more potential energy on the platform than in the water. And then diving converts potential energy to kinetic energy because now the diver is moving. So then as the diver climbs back up, climbing up converts the blank energy of muscle movement to potential energy. So that's going to be the kinetic energy of muscle movement to potential energy. And then this last box here, a diver has blank potential energy in the water than on the platform. That's going to be less potential energy. So entropy is really the relative amount of disorganization. And again, in another class, you might have a more refined definition of entropy, but that's one we can work with for our class for now. So the only way to maintain or bring about order is to add energy. If we look at this pile of bricks, like this first pile of bricks, let's say just got dumped off of a truck, in order to get this pile of bricks to look like this neatly stacked pile of bricks, we would need to add energy in and organize that. So energy provided by the sun allows plants to make glucose from the more disorganized water and carbon dioxide. So plants doing photosynthesis take energy in and create a larger, more organized molecule. Not all of the energy from the sun is transferred into the carbohydrates in the plant. Some of it is, quote, lost to the environment as heat. It's not really lost. That's actually really important for us. But not all of the energy and light makes it into carbohydrates in plants. So individual cells and whole entire organisms can create ordered structures from less organized starting materials by adding energy in. So let's review. Which of these pictures has more entropy? Don't worry, this isn't from my house. This is just a random picture from somewhere in the internet. Although honestly, at any time, anything could look either way. So this second picture is more disorganized, so it has more entropy. And to go from the second picture to the first picture, which is more organized, we'd have to add energy in. So that brings us to endergonic and exergonic reactions. Endergonic reactions describe a reaction that requires a net energy input, so energy is stored in the products. Exergonic reactions, on the other hand, describe a reaction that releases energy by breaking bonds. So free energy is the portion of a system's energy that can do work. It's a measure of the system's instability, its tendency to change to a more stable form or state. Spontaneous reactions can be harnessed to perform cellular work, and during a spontaneous change, free energy decreases and the stability of the system increases. So in this picture here, everything on this kind of top row has more free energy, it's less stable, and it has greater work capacity. So our diver has more potential energy here. These molecules that are close together may bump into each other and move further apart, and this larger organic molecule has more energy between those chemical bonds. So when the diver jumps off, there's less free energy available. The diver in the water is more stable than on the platform. There's less work capacity there. These molecules being further apart in the second column are less likely to bump into each other, so there's less capacity for work, and we've released energy from these chemical bonds in this chemical reaction, so there's less work capacity there as well. So let's review. Which of these pictures with the person with the ball represents kinetic energy? So that's going to be the first picture here because the ball is moving. In the second picture, that's going to be potential energy because of the position of the ball. For number 4, which graph, A or B, represents an endergonic reaction? So an endergonic reaction is a reaction that requires an energy input. So B is going to be an endergonic reaction because there's more energy stored in the products than there were in the reactants, and energy is input into the reaction. A is an exergonic reaction. So living things are not necessarily in equilibrium because living things are open systems. We take material in, we remove waste from our bodies, and that helps control our reactions. Oftentimes, energy releasing exergonic reactions can drive energy requiring endergonic reactions. And oftentimes the energy releasing reaction is ATP hydrolysis. Remember, hydrolysis is breakdown. So we're saying when we break down ATP, that releases energy. So these are a couple kind of cheesy pictures here, but in this first picture, this wagon going downhill is pulling the second wagon up the hill. So this wagon going downhill is kind of like the energy releasing reaction that's pulling the wagon uphill because this requires energy. This second picture, there's a few things a little bit wrong with it. Like that is a very sturdy wagon if it's hit by a cannonball and okay. And like the physics of our cannonball aiming isn't necessarily technically going to hit the wagon. But if we ignore that, what this picture is trying to show us is that the transfer of a molecular group, like the transfer of a phosphate group from ATP to another molecule, could drive that reaction. So we've talked about ATP a few times already, but ATP has adenine and then ribose, which is a 5-carbon sugar. This part doesn't change, and then there's 3 phosphate groups in ATP. 1, 2, 3. Oftentimes they simplify it and just show you little P's here for the phosphate groups of ATP. So the phosphate groups can break apart and release energy. So if we have 3 phosphate groups, it's adenosine triphosphate. The T stands for tri. If there are 2 phosphates, that would be adenosine diphosphate. The D stands for di, which is 2. And ADP can break apart, and if there's only 1 phosphate group left, it's AMP, which the M stands for mono, so adenosine monophosphate. So ATP is really kind of the energy currency that all cells use because the hydrolysis of ATP releases energy. It's ultimately because of the repulsive force of the 3 negatively charged phosphate groups. This bond between the phosphate groups is a high energy bond, so when we break that, that releases energy. So ATP can be used to do all kinds of work in the cell. For example, it could be used to do transport work. So this picture up here is showing active transport, where we're having a phosphate group bind to a transport protein and move this solute against its concentration gradient. This is showing ATP powering a motor protein on a vesicle as that vesicle is propelled along the cytoskeleton there. So ATP can be used for mechanical work, transport work, and even chemical reactions. So the hydrolysis of ATP, again, is when we add a water in and break apart a phosphate group from ATP. So we get ADP and a free phosphate group, and energy is released. We can rebuild that ATP by adding energy in and linking another phosphate group to the ADP, so we get an ATP, or adenosine triphosphate. We'll talk more specifically about the regeneration of ATP a bit later, but basically ATP is a renewable resource, and the energy to rebuild that ATP comes from catabolic reactions in the cell. What's catabolic mean again? So catabolic is breakdown. So when we break down larger molecules, that releases the energy we need to make ATP. So there's 2 kind of big ways we can do this. We can do substrate level phosphorylation, where the energy stored when phosphate is added to ADP comes directly from breaking down an organic molecule. Or we can do oxidative phosphorylation and chemiosmosis, which is a little bit more complicated. And that's when we have this proton or hydrogen ion concentration gradient, and those hydrogen ions or protons flow through this kind of spinning molecule here, or spinning protein complex called ATP synthase. So these hydrogen ions flow through ATP synthase, kind of like how wind goes through a windmill or water goes through a water wheel, and that powers the production of ATP from ADP, so adenosine diphosphate, and a phosphate group. We get adenosine triphosphate, or ATP. So let's review. Number 5, the energy to synthesize ATP comes from chemical reactions that are what? So the energy we need to make the ATP comes from exergonic reactions. Making ATP requires energy, it's endergonic, and we get that energy from exergonic reactions. So 5 is A. Number 6, ATP hydrolysis provides the energy to drive cellular processes that are blank. So hydrolysis, again, is break apart or break down. So we're saying we break down ATP, that releases energy, and that energy drives processes that are endergonic, or energy requiring. So just because a reaction is spontaneous doesn't necessarily mean it will occur quickly. All reactions require an input of energy to start the reaction. We call that energy input the activation energy. This activation energy, we can think of it as allowing molecules to get close enough to cause bond rearrangement to occur. Oftentimes in a lab setting, in order to overcome activation energy, we would add large amounts of heat. For example, in that organic molecules lab, we had to add the Benedict's reagent tubes and then anhydride reagent tubes to a hot water bath in order to make that reaction occur in a more timely fashion. You can't really boil living things though, right? They don't generally stay alive when you do that. So living things, we usually don't have a huge amount of heat added to overcome the activation energy. Instead, we use enzymes, which are biological catalysts. And a catalyst is just a chemical agent that speeds up the reaction without itself being consumed by the reaction. So enzymes are biological catalysts. Most of them are proteins for now. In this chapter, we'll go ahead and say they're proteins. And the shape of the enzyme stabilizes a temporary association between substrates. So the enzyme speeds up the rate of a chemical reaction, but the enzyme itself is not changed or consumed in the reaction. And enzymes only work on specific substrates. So enzymes only react with specific reacting material. Usually in enzymatic reactions, enzymes are named for the name of their substrate with the ending "ase" added to the end. So in our reaction below, which of these is the enzyme? This reaction has lactose plus lactase plus water, and we get lactase, glucose, and galactose. So we're looking for the thing with the "ase" ending. So lactase is our enzyme. Another hint that lactase is the enzyme is it's on both sides of the chemical equation there. So again, that activation energy is the energy required to start that reaction. And with enzymes, that reduces the activation energy. So enzymes increase the rate of the chemical reaction. They make starting the reaction easier. Some enzyme terminology. The enzyme is the thing that's bringing our substrates together. Substrates are the new term for reactants in an enzymatic reaction. So substrates bind to something called the active site on the enzyme, and an enzyme substrate complex is formed. And there's a couple different models used to describe enzymatic reactions. The induced fit model says that the enzyme's shape temporarily changes when the substrate binds to it. And there's another kind of metaphor used to describe enzymatic reactions where we talk about a lock and key metaphor, where the substrate and the enzyme fit together like a key fits into a lock. So this picture here kind of takes us through an enzymatic reaction. We've got our substrates. They bind to the enzyme's active site. An enzyme substrate complex is temporarily formed. Those substrates are converted to products. The products are released from the enzyme, and the enzyme is available to bind to new substrates. So there's some things that can change the rate of the enzymatic reaction. If we increase substrate concentration, that increases the rate of the enzymatic reaction until all the enzymes are fully occupied by substrate. When that happens, the enzymes are said to be saturated. When the enzymes are saturated, they can't accept more substrates, so they can't go any faster than they're already going. Another thing that can alter the rate of an enzymatic reaction is temperature. Generally speaking, as temperature increases, the rate of a reaction increases. However, since enzymes are proteins, their shape determines their function, right? And at high temperatures, proteins can be denatured. If the enzyme is denatured, it will no longer be able to bind to the substrate, so the rate of the reaction would decrease. The same sort of thing with pH. There's optimum pHs for different enzymes, and if the enzyme is outside of its optimum pH range, then the rate of the reaction will decrease. With inhibition, this occurs when the active site of the enzyme is prevented from combining with its substrate. So there's competitive inhibition and non-competitive inhibition. In competitive inhibition, the inhibitor is binding to the same active site as the enzyme, so they are actively competing for the active site. In non-competitive inhibition, the inhibitor is binding to an area other than the active site on the enzyme. So they're not directly competing, but both cases, the substrate is blocked from binding to the active site of the enzyme. So what are some things that could alter an enzyme structure, and how would this impact its function? So too high of a temperature could denature the enzyme structure, and that would reduce the rate of the enzymatic reaction. Also, the wrong pH could denature the enzyme and lower the rate of reaction, and inhibitors could alter the rate of the enzymatic reaction by either binding to the active site or binding to another part on the enzyme. Number 8, at enzyme saturation, how can reaction speed be increased? So remember, at saturation, that's when all of the enzymes are fully occupied by substrate. So there's more substrate available than enzymes to do work on those substrates. So to increase the rate of reaction when the enzymes are all saturated, you'd have to add more enzymes in. So cofactors and coenzymes, that root word co means together. So cofactors and coenzymes are going to work together with enzymes. So cofactors are non-protein enzyme helpers, and an organic cofactor is called a coenzyme. We're going to really focus more on their function. A coenzyme will easily accept or lose electrons, so they can act as an electron carrier. This is a necessary intermediate when we're going from organic molecules to energy. We're going to talk about NAD, FAD, and NADP in the next 2 chapters. So NAD, FAD, and NADP are coenzymes. In living things, there's actually multiple reactions going on at a time. So a metabolic pathway, or a biochemical pathway, begins with a specific molecule and ends with a product several steps later. And each step is catalyzed by a specific enzyme. So the product of one reaction becomes the substrate for the next reaction, and so on and so forth. So what would happen if an enzyme was missing? Let's say in our example above, if enzyme 2 was missing, what would happen? If enzyme 2 was missing, we wouldn't get C, right? Product C. So we wouldn't be able to do step 3. So we wouldn't be able to get our final product if an enzyme was missing along the way. So we can control an entire metabolic pathway with feedback inhibition. Feedback inhibition is when the end product of a metabolic pathway feeds back to an earlier reaction and inhibits an enzyme earlier on in the pathway. So feedback inhibition prevents a cell from wasting chemical resources by synthesizing more product than is needed. So in this example, isoleucine is the end product, and isoleucine actually feeds back and inhibits the first enzyme in this metabolic pathway. So isoleucine can bind right here and inhibit enzyme 1 from progressing on the pathway. So let's relate the following terms. In the top row, we've got dehydration synthesis or condensation reactions, anabolic reactions, endergonic reactions, and it says may be coupled to ATP hydrolysis. So if we look at our first term, dehydration synthesis or condensation reaction, that's where we take a water out and link molecules together. So we're building something there. Anabolic reaction, that is a metabolic pathway where we build something. Endergonic reactions, that's when we add energy in and build a molecule. So really those first 3 terms, those are kind of a different lens to look at the same thing. All across those 3 terms, we're building something. But the first one, we're talking about specifically how we're doing it. We're taking a water out and linking monomers together. The second part says, oh, we're building something. The third part, endergonic reactions, that says it requires an energy input. And the last part says may be coupled to ATP hydrolysis. ATP hydrolysis is the breakdown of ATP. When we break down ATP, that releases energy to build molecules in this case. That's not the only thing it does, but that's one thing it could do. In the bottom row, we've got hydrolysis, catabolic, exergonic reactions, and then it says may be coupled to ATP synthesis. So hydrolysis, that's where we take a water out between 2 monomers in a polymer and separate the polymer into the monomers. So we are breaking things apart. In catabolic reactions, that's broadly the reaction where we break things apart. Exergonic reactions are energy releasing reactions, and we release energy by breaking things apart. And that energy that's released may be coupled to ATP synthesis or the production of ATP. So, what are 3 other terms that could be used to describe a dehydration synthesis reaction? Well, we could say it's a condensation reaction. We could say it requires an energy input, so it's an endergonic reaction. Or broadly, we could say it's an anabolic reaction. Number 11, oxidative phosphorylation and chemiosmosis are one way to produce ATP. What is the other type of phosphorylation? That is called direct or substrate level phosphorylation. So we made it to the end. We talked about metabolism, which is all of the chemical reactions going on in an organism. Energy is the capacity to do work. We talked about thermodynamics, which is energy transfer from one form to another. We talked about endergonic and exergonic reactions. So endergonic reactions require a net energy input. Exergonic reactions release energy. So it takes energy to do work, and oftentimes in living things, the energy comes from the hydrolysis of ATP. ATP has 3 phosphate groups. When we hydrolyze it or break it apart, we release a phosphate group and we get a DP or adenosine diphosphate. We could break it apart one more time and go from adenosine diphosphate to adenosine monophosphate with 1 phosphate group. We talked about enzymes and how enzymes increase the rate of chemical reactions. In an enzymatic reaction, the substrate binds to the enzyme's active site, and you temporarily get an enzyme substrate complex, and then products are released, and the enzyme can go back to work again and bind more substrates. We can alter the rate of enzymatic reactions by increasing the temperature. Generally, that increases the rate of the reaction unless we increase the temperature too high and we denature the enzyme. There's also optimum pHs for enzymatic reactions, and if the pH is too high or too low, that could denature the enzyme as well. There's an optimum concentration for enzymatic reactions. If the concentration of substrate is too high, then the enzymes will be saturated, and adding more substrate would not increase the rate of the chemical reaction. Enzymes can be inhibited by either competitive or non-competitive inhibitors that affect the ability for the substrate to bind to the enzyme's active site. We can also control multi-step metabolic pathways with feedback inhibition, where the final product of that pathway feeds back to an earlier enzyme and inhibits that enzyme from binding with substrate.